Hello, students. All right, we are going to continue with 11.1 monoclonal antibodies. And you might have heard of these lately because they are in the news a lot as a COVID-19 treatment. So the essential understanding is that fusion of tumor cells with antibody-producing cells creates a hybridoma. And it's these hybridomas that are then going to give rise to the monoclonal antibodies. So monoclonal antibodies are produced by hybridomas. So what are these monoclonal antibodies? These are antibodies that are artificially derived from a single B cell clone, hence the name monoclonal. They come from a single B cell clone. So let me move this up here so you can see the picture. So typically, these monoclonal antibodies are derived from an animal. And in this particular case, it's a mouse. So just quickly, let me move here. So there are different examples of therapeutic monoclonal antibodies. They can come 100% from a mouse. So they're called murine anticlonal antibodies, and they have 0% human antibodies in the mix. These are called OMAB. And then we have chimeric monoclonal antibodies, and they have about 65% human monoclonal antibodies, and they're called Zymabs. And then humanized, which are greater than 90% monoclonal antibodies, and they're called Zumabs. And then we have 100% human anticlonal antibodies, and they're called UMABs. So these UMABs are the ones that are being used for COVID-19 monoclonal antibody treatment. And notice that they have a low potential for immunogenicity, which means that you're not going to have an adverse reaction to them because they're 100% human. So let's move back and see how they're produced. So like I said, typically they're murine, meaning they come from a mouse. So what happens? A mouse is injected with an antigen, and this antigen produces specific antigen-specific plasma cells, right? Now, remember that these plasma cells are going to in turn produce antibodies and memory cells. But how do you get a specific antibody from the specific plasma cells? So the plasma cells are removed from the mouse's bloodstream, and then they're hybridized. Here's what makes them different. They're hybridized, meaning that they are fused with tumor cells. And these tumor cells come from particular immortal cell lines. Now remember that these immortal cell lines, you might have heard The Immortal Life of Henrietta Lacks, they come from some form of aborted fetuses, right? And these immortal cell lines are just kept over and over, reproducing for 10, 20, 30, 40 years. So some of these immortal cell lines that were derived from aborted fetuses have been immortal for over 60 years now. So they have endless divisions. The immortal cell lines that were retrieved from Henrietta Lacks, they were from a cancer that she had, cervical cancer that she had, and we're still using immortal cell lines from Henrietta Lacks' tumor. So anyways, you take these plasma cells that are made by the mouse and you hybridize them with these tumor cells. So now you end up with a hybridoma, and this hybridoma is capable of synthesizing large quantities of monoclonal antibodies. And they're called monoclonal because they're specific to these plasma cells that were induced by this specific antigen, and they're all clones. So they're going to target specifically, right, specific immunity, they're going to target specifically this antigen here, which has induced the whole immune reaction. So for COVID-19, for example, they're not using mice plasma cells. What they're doing is they're trying to retrieve, and this is relatively new science within the last 10 years, they retrieve the plasma cells from people who've already been exposed to COVID-19 and whose plasma cells were derived from the antigenic immunity from COVID-19, right? So they take these plasma cells, they hybridize them with an immortal cell line, and they make this monoclonal antibody cocktail that then can be infused into a person who has COVID-19, and this could reduce the symptoms of COVID-19. All right, so we know that monoclonal antibodies can be used for therapeutic treatment, as we saw just recently with COVID-19, but also in clinical detection of the disease. So here's the application. Monoclonal antibodies to HCG are used in pregnancy test kits. So an example of a therapeutic use involves the use of antibodies to treat rabies, to treat COVID-19. But what you are responsible for knowing in this application is how it's also used to detect pregnancy. This is what we're going to spend some time talking about right now. So we already talked about these different examples. All right, so how are monoclonal antibodies used to detect pregnancy? So what they detect is the presence of human chorionic gonadotropin, or HCG, that is found in urine of pregnant women. So HCG is a hormone that is produced by women during fetal development. So it is present in the urine of females that are pregnant, therefore indicating pregnancy. So pregnancy tests, like the ones you buy at Walgreens and CVS, they work via a process called ELISA, or enzyme-linked immunosorbent assay. And this is used to identify the presence of these HCG hormones via a color change. So let's see how these tests work. Okay, I'm actually going to leave it up to there. All right, so free monoclonal antibodies specific to HCG are conjugated to an enzyme that changes the color of a dye. So here we see the pregnancy stick, right? And here we see that there is a portion of the pregnancy stick that you pull out, and this is called the reaction site. And in this reaction site, you have these free antibodies, these free monoclonal antibodies that are specific to HCG. So they're going to trap HCG. They're going to react with this dye. So when the monoclonal antibody comes into contact with HCG, the dye is going to change colors. Okay. Then you have a second set of monoclonal antibodies that are also specific to HCG that are immobilized to the dye substrate. So in this particular region right here, you have a fixed antibody that traps HCG and also contains a dye substrate or a dye component. And then you have a third set of monoclonal antibodies that are going to bind to any unattached enzyme-linked antibodies. And that's what functions as a control. So this is this third site. So we have the first site here, the second site here, and the third site here. This corresponds to these free antibodies. This corresponds to the fixed antibodies. And this corresponds to fixed antibodies that can trap any free antibody. Okay? So let's move down here and see what happens. So in a pregnancy test, this is the reaction site, or where you pee, right? So when urine reaches this area, this reaction site, what happens? Well, these free antibodies are able to move along the stick, right? So they move along the stick, and if these free antibodies are of a pregnant person, right, these free antibodies have been able to trap the HCG present in the urine. So as these free antibodies move along the strip, when they reach this second test site, what happens is they also react with these fixed antibodies that cannot move, and these fixed antibodies also trap the HCG found in the urine. And they react with the dye, so then this line turns blue. and this line appears. So if this line here appears, that means that you're pregnant. That means a positive result. Okay? Now, these free antibodies here that have trapped the HCG, they keep moving, right, down the stick here. And they also react with these free antibodies and these fixed antibodies. And these fixed antibodies are able to trap any type of protein or enzyme. So it also turns blue, and it's just a control site. It means that the test is working. Okay? So if you're pregnant, you're going to get a positive result by seeing two lines. This line here and this line here. This one being the test site and this one being the control site. Now, if a woman who is not pregnant, so if you're not pregnant, you're not going to have HCG hormones. So if a woman that's not pregnant, if the woman pees on this stick, there's not going to be any trapped HCG. So then the free antibodies are just going to move down the stick. Nothing is going to get trapped here in the fixed antibody that traps HCG. So the urine is just going to continue moving. This line is not going to turn blue. So the pee is just going to continue moving. The urine is just going to continue moving. And then it's going to reach the control site where there are fixed antibodies that is just going to trap any type of protein-like antibody because this is the control site. And this is going to turn blue, right? So that means you're only going to end up with one line. So one line means a negative result. It means the test is working, but you are not pregnant. So again, two lines is a positive result, means you're pregnant. One line only in the control site means a negative result. It means that you are not pregnant. I'm also going to attach a video that shows you how the antibodies are moving down the stick, also to help you with this concept. All right, that is it for monoclonal antibodies.